Dear students, the topic of discussion today is gene therapy. Gene therapy is an experimental technique that uses genes to treat or prevent disease. Replacing a mutated gene that causes disease with a healthy copy of the gene is the main objective of gene therapy. Inactivating or knocking out a mutated gene that is functioning improperly, and in this place, introducing a new gene into the body that help fight the disease is the fundamentals of gene therapy. How does this work? Mutated gene always gave rise to faulty protein, whereas the correct gene gives a functional protein. Gene does not function directly, but as always, it is to be combined with the carrier, and the carrier here is a vector. Which is a vector we use? It is usually viral vector. Why? Because viruses are often used as vectors because they can deliver the new gene by infecting the cell. The examples of the viruses used as a carrier for the gene delivery is retroviruses, where they integrate their genetic material that includes a new gene into the chromosome of the human cell. The next example being adenoviruses. This introduces the DNA into the nucleus of the cell, but the DNA is not integrated into the chromosome of the cell. The third virus being adeno-associated virus. These are the viruses which mainly infects humans and primate species, but they lack pathogenicity, which is an advantage. The fourth example of the virus, which can be used as a vector for the gene delivery, is herpes simplex virus, where this is a natural human pathogen which replicates in epithelial cells. It is having the largest cloning capacity of all the vector, where they can carry the gene of 150 kilo base size. This vector carrying the correct gene can be injected or given intravenously directly into specific tissue of the body where it is taken up by the individual cells. On the other hand, a sample of the patient's cells can be removed and exposed to the vector in a laboratory setting. The cells containing the vector are then returned to the patient. The treatment is successful. The new gene delivered by the vector will make the protein or will make up the functioning protein. This is a diagram which speaks about the types of gene therapy, in vivo gene therapy and ex vivo gene therapy, depending upon the procedure which is used. Now, this is a diagram which speaks about both ex vivo as well as in vivo. The cells having the mutated gene are removed from the patients, and to this the cells, and in the cells which are having the faulty gene is replaced by the correct gene and cultured in the laboratory. It so happens the transfer of the correct gene can be taken by the cells, and the cells are called as positive cells, where these positive cells are later amplified in the laboratory conditions using specific screening and selection methods and the medias. This amplified X cells, which are now the correct cells having the correct gene, can once again return back to the cell, to the individuals in the form of IVs or intramuscular injection. Thus, this diagram speaks about both ex vivo as well as the inclusion of in vivo gene therapy. This is how it happens, a procedure wherein we will be using a vector, a carrier, where in this vector we have the modified gene. We have the modified gene which indicates that the faulty gene is removed and the correct gene is put. Later, this vector is usually given in the form of IV or IM, as this vector is injected into an individual's. These vectors, as they enter into the cells, they get packaged in the form of vesicles. As the vesicles near the nucleus of the individual, now what they do is that the vesicles breaks down, releasing the vector. Vector now injects a new gene into the nucleus where the cells are programmed to synthesize a protein because there is a new gene, in other words, a correct gene. The procedure here seems to be very easy, but practically there is lots of barriers involved. Practically, there are lots of hindrances which are as to be faced, or the challenges are many. So we will be speaking about what are the barriers to the gene delivery, or what are the challenges one is going to face to bring about the successful gene delivery in a case of treatment. But before that, let us speak of the types of gene therapy depending upon where it is performed or which cell it is performed. And thereby we have two types of gene therapy. One is somatic gene therapy and the other one is germline therapy. Now, the major difference lies like this. In the somatic gene therapy, the genes are transferred to the somatic cells. They affect only the targeted cells in the patients and they're not passed on to the future generations. Appropriate and acceptable for many disorders, and it is being also implemented for one of the diseases or few of the diseases such as cystic fibrosis, muscular dystrophy, cancer, and certain infectious diseases. On the other hand, we have the germline gene therapy, wherein this is being done on the sperms or the eggs. The sperms or the eggs are modified with the introduction of the functional genes, where these genes are integrated into the genome, thereby allowing the therapy to be heritable and passed on to the later generations. And they're highly effective in counteracting the genetic disease and the hereditary disorders.